Alright, so let's move on to the fourth video, which is on the vectors itself. So I'm dividing the vectors into viral and non-viral vectors, so we will cover the non-viral vectors next week. But first, the viral vectors. So for the viral vectors, I'm actually only going to give you 5 examples. There are a lot of viral vectors that could be used for gene therapy. I'm using the ones that has been proven, that has been studied in details. You can find papers on them only. So for gene therapy, using the viral vectors, so the virus is of course first and foremost modified to become a vector to engineer your genes, because if not, then it will be detrimental or it could be infectious to the person handling it and of course the patient later on. So the viral vectors have to have a wide range of gene packaging and cell entry mechanism. Again, remember back on what you have learned in gene therapy or cell molecular or cellular biology, because there are a few types of vectors. So among the two, viral vectors is more effective than a non-viral vector because it has a very good gene packaging and also gene delivery mechanism. It also has a certain mechanism to evade the attack from the immune system. So the criteria of a good viral vector is it should be safe. Well, you will see some of the examples I'm going to give you is actually quite dangerous, but we can make it safe. It should not integrate into the host genomic DNA. So if you remember on the diagram just now, only the therapeutic agent is being delivered. The host remains outside, not into the nucleus. It should not replicate because then it will basically evade and overcome the host system. It should be genetically stable. It should be easy to modify, and it should provide sufficient expression of transgene. Now, we know that in nature, viruses attack hosts to produce its own components, but it could also carry certain extra genes. So viral vectors are usually packaged into special cell lines to produce replication defective viral vectors. So simply said, in theory, insertion of missing gene into the virus will cause the virus to spread the gene into the patient's cells upon infection. But inside the patient, the missing gene is actually being replaced by the transgene that is being carried by the virus, and of course, hopefully, the disease is cured. So for therapeutic use, the viral genome has to be altered definitely to make sure that it's safe and it does not infect or invade the host cell. This is done by removing essential viral genes to restrict the replication. And in place, of course, therapeutic gene is added, or therapeutic genes are added. So this is more or less how the packaging system or the mechanism of the virus is being utilized. So what happens again is that again, in its nature, or the normal wild type virus, okay, they basically have their own genome, whether it's in RNA or DNA version. So they make their own plasmid and also viral structural genes, and this is being packaged and whatnot. So this is being utilized. But instead of having the viral genome, so your therapeutic gene is being incorporated into the viral genome, and it will be replicated, okay, and of course being packaged by the mechanism of the viruses, depending on the types of viruses. Alright, so basically what happens is that when you modify the viral vectors, it usually, or the viral replication genes are usually deleted or basically turned off. So because it is being because it doesn't have its own original missing replication genes, so it has to be cultured in a transcomplementation cell line to make sure that it could replicate your transgene later on. So after you have done the complementation, you can basically do the purification, and the virus could then be administered for therapeutic purposes. So before exposure, it may infect and deliver therapeutic genes, but not to reproduce. This is just a precautionary measure to make sure that the virus property is basically totally devoid in the vector. And since viral replication genetic information is of course missing, as I said earlier on, so transduced cells will not be able to replicate the virus. So replication of the transgene is actually being done by incorporating another version of replication mechanism. And these are the types of viruses used as vectors in gene therapy. So the first one, like I said, we have 5. So the first one is adenoviruses. So for those of you who don't know about adenoviruses and retroviruses are the most commonly used virus as vectors, not only for gene therapy, but a lot of other purposes as well. So for adenoviruses, its genome is made of double-stranded DNA, which encodes lots of genes. So in terms of its structure or morphology, it consists of icosahedral capsids with 12 ventricles and 7 surface protein. You can see over here, okay, 7 ventricles. Okay. So the virion, okay, basically is non-enveloped, spherical, and have the size of about 70 to 90 nanometers. So that means that it is basically very small in nature. So what happens again is that because it's actually very small in nature, so it would be very hard to be detected, and hence it couldn't be seen with naked eyes. But this is also one of the good things about the virus. So in terms of adenoviruses, its genome encodes about 30 proteins, and the transcription of the genome occurs in 3 stages, either immediate early, or late. So once infection has taken place, the DNA is not incorporated into the host DNA but left free in the host nucleus. So this is the advantage of adenovirus. So the extra genes from the virus will not be replicated in the cell, so it will not be integrated into the genome. So in normal or in nature, if anyone is being infected by adenoviruses, it could cause common cold or sometimes intestinal and eye infection. And this is, like I said, how it looks like in nature. So this is the icosahedral, kind of like has 6 sides, okay, but 3D structure. So this is how it looks like. So it has basically the 7 surface proteins and whatnot. Okay, so it's actually very small. Okay? This is the TM photograph of the adenovirus. So the second one of the virus is the retrovirus, like I said early on. So just like adenovirus, retrovirus could basically create double-stranded DNA copies, but it actually has RNA genomes. Okay, so it has 2 copies of RNA genomes. It doesn't have DNA genomes, it has RNA genome, but it could be able to create double-stranded DNA copies, kind of like cDNA. So the RNA genome is packaged in a protein capsid surrounded by a lipid envelope, and it contains reverse transcriptase enzyme. So that's why it is able to create double-stranded DNA copies. So in terms of its morphology, it's spherical in shape with a diameter of about 80 to 100 nanometer, slightly bigger than adenovirus. So the internal structure comprises mainly the product of the GAG gene. For example, it has the LTR for integration of virus into host genome. It has the GAG, okay, which correspond to core encapsid protein to make sure that it is packaged thoroughly. It has the POL that codes for RT enzyme, reverse transcriptase. It has the ANV that codes for GP120 glycoprotein for the packaging system. And it has the TAX or RAX that codes for transactivation factor. So once infection occurs, the RNA is actually being injected into the cytoplasm with the RT enzyme, okay? And the cDNA will be produced containing viral genetic instruction. So an example of retrovirus is actually HIV, or human immunodeficiency viruses. Right, so this is the structure of the retrovirus. It's basically kind of like spherical in shape, slightly bigger, but it basically has lots of basically protein lipids over there, as you can see. So when it has lots of this lipids, what happens is that it could attach to any surface easily, making it one of the most, I would say, dangerous but also an effective vector to be used for delivering transgene. So the next one is lentiviruses. So this is actually a subclass of retroviruses. It could infect proliferating and non-proliferating cells. So the mechanism of lentivirus rely on active transport of your target cells, and it basically the good thing about lentiviruses, okay, is that it could infect non-dividing cells. This would be useful especially if the gene or defective gene is basically located in the non-dividing cells. So for lentiviruses, usually it is being constructed as a hybrid vector from non-human with similar approaches used in HIV-derived vectors. And this vector efficiently transduces non-dividing tissues of rodents, humans, and other species for ex vivo. In vivo hasn't been tested, or I actually haven't come across any paper yet. So there is an obligatory RNA step in retrovirus life cycle that restricts the viral genome and the exploitation of the lentiviruses. It is only a subclass of retroviruses, so that means that it doesn't have the full function of the retrovirus. But again, the good thing about lentivirus is that it could infect non-dividing cells. So basically, the application of lentiviruses is basically it could be used, okay, or could be applied for transduction of hematopoietic stem cells. So this is how lentiviruses looks like. This is the actual TM micrograph. So it is quite similar to retroviruses because it's actually just a subclass of it. In terms of the size, it's more or less similar. It has quite all the machinery, similar machinery as to the retroviruses as well. So the fourth virus, the vector, is adeno-associated viruses. So from the name, you can guess for retrovirus, we have a subclass. So for adenovirus, we also have a subclass in terms of adeno-associated viruses. So as compared to the adenovirus, adeno-associated viruses, or AAV, basically has small single-stranded DNA as its genome. So it basically, its specialty is to insert its DNA in chromosome 19 in human. Why chromosome 19? Google up about it. I'm not just going to give you everything. All right, so in nature, it is also known, one of the most common AAV is also known as human parvoviruses, requiring a helper vector virus to mediate infection. If anyone has ever heard of parvoviruses, and probably from animals, dogs or cats, so it's actually more or less similar mechanism. It could be detrimental, the effect. So the viral genome consists of two genes, with each producing multiple polypeptides, wrapped for viral genome replication, and capped to encode protein to direct in the packaging and also the infection. So the vectors can be produced by adding separate plasmid flanking therapeutic genes and adding of helper adenovirus. So basically, it could be usually put here, somewhere here, or you can put it somewhere here, but it will be tricky. So transduction of vectors can occur through episomal transgene expression and random chromosomal integration. So it is especially useful for in vivo application, as the vector particles can be delivered to many different organs. But the vector can only be integrated into the genome, and production in large quantities is not easy. They have tried this. AAV, because of the small size as well, and basically restrict production in large quantities. So this is how AAV looks like. So it's actually smaller in size as compared to the adenoviruses. Quite similar, but it's not really icosahedral. It's more towards kind of like spherical in shape. And the last one, again, the last vector that I'm just going to put here for you to see on the differences is the herpes simplex viruses. So I know some people say herpes, some people say herpes, but the actual or the correct pronunciation is actually herpes. But it doesn't really matter even if you pronounce it as herpes. So herpes simplex viruses, basically in nature, it causes common cold, or cold sore. So cold sore is whereby kind of like when you see there are some form of inflammation around the corner of your mouth. You can just Google up and you will see the picture. It's actually not nice to see. So usually if you have cold sore, and once you have been infected by herpes simplex viruses, you will have it for life. You will not be able to cure yourself of it. But it doesn't really affect you that much unless you are being attacked by a very vicious strain of the herpes simplex virus. Okay, moving on. So it has, in terms of its genome, double-stranded DNA, and it infects the neuron. So that's why herpes is actually quite dangerous. So it has a large genome with a large number of genes, but so far I think people have studied about 50 to 70 types of genes. The exact number hasn't been said. So herpes simplex viruses able to persist after primary infection in human, and of course it is useful for in vivo because it could basically hide itself or basically prevent itself from being detected by the immune system. But of course it has a bit of a disadvantage in terms of the herpes virus vectors should not be reactivated and produce infection, because it could produce infectious virus or cause other diseases, for example cold sore. So there are various limitations to this vector, making it the least popular choice, because in nature itself it has infected a lot of people. I have actually one of my students being infected by herpes simplex. She has cold sore up until now. So one of the limitations is, among the limitations are long-term transgene expression in certain tissues, Vector targeting is actually not very easy. Complex attachment and entry mechanism, lack of experience with recombinant herpes viruses in patients because it is quite a virulent strain, restricted target cell spectrum, transduction dependent on cell division, which means that it could infect non-dividing cells, and basically because it is a common virus that infects our bodies develop antibodies against herpes. So that means that to use herpes as a virus vector, probably it will be attacked by the immune system. So this is how the herpes looks like. So it looks like this. So that's why if you are being infected, when you have a cold sore around your mouth or sometimes at your back, behind your back, your body, you will have this kind of formation on the skin, kind of like lesion. Alright, so I think those are the viruses that I'm going to discuss. So next week we're going to discuss on the non-viral vector, and be prepared for yourself. I will also remind you guys on Tuesday that next week we're going to have a quiz during lecture. Be prepared on these vectors.